All right, let's look at two more cestode case studies. And in this one, we have a 20-year-old man sitting at his desk when he sneezed and noticed a small object fall from his nose onto his desk. That could be any one of you. Imagine how freaked out you'd be. I'd be freaked out for you. He claimed the object was moving on the desk, which is strange because actually flatworms can't move because they don't have musculature, but whatever. He took the object to his healthcare provider, who thought that it was a kind of worm, and the object was about 0.7 centimeters by 0.2, somewhat flat, whitish in color. It was sent to the pathology department where it was sectioned and stained. So what I want you to look at here is that this is not like a typical stool sample, right? This is actually a section of a worm that has been cut and stained. It's kind of almost like a pathology prep, right? So the pathologist did look at the prep and saw several images. You can see them here. And what I'll tell you is the one that really counts is the one in the bottom right, figure D. So what do you think this might be and why? And remember, what you're looking at in figure D was inside of the worm. So maybe pause for a second and see if you can get a rough guess before I give you some more hints, right? If we look at figure D in a different light, it kind of looks like this. There are radial striations along the outside. You can see a hexacanth, it looks like a freaky alien, but you can see a hexacanth embryo in here with the little hooklets. Kind of freaky. And it's stained kind of a dark brown because it absorbs the stercobilin from the feces. Now the catch here is that this looks a lot like two different cestodes, and it actually looks like a nematode too, but we're going to hold off on that until next week. Could be one on the left or on the right. So these are proglottids from two different cestodes that both produce the same eggs. Do these proglottids lend you towards any particular cestode? And then here are scolices of two different organisms, and this egg comes from either Taenia solium on the left or Taenia saginata on the right. Both of these tapeworms produce the same egg, right? Their eggs are identical. And so again, if I give you the egg, I'd have to give you context or I'd have to accept multiple answers. How are you going to tell them apart beyond that? Well, looking at the proglottids, what you will notice is that there are more uterine branches in Taenia saginata. So if this is the uterus, there's more fine branching here. Whereas in Taenia solium, it's thicker, and by default, there would be fewer. If you look at their scolices, they're going to be different than Diphyllobothrium latum because they have 4 sucking discs. They both have 4 sucking discs. And you're going to see there is a rostellum, and you can't really see it here for saginata, but it has a rostellum, which is kind of that protrusion. Only Taenia solium has an armed or armored rostellum. So you can see the hairs here. Those are actually little hooks that can anchor into the wall. On the left, Taenia solium is the pork tapeworm. On the right, Taenia saginata is the beef tapeworm. And the good news is that their life cycle is pretty identical, minus one little thing, and I'll point that out. So this is kind of the key identification stuff. If we look at the life cycle, we'll start at the top. Let's start with the infective stage here. Notice we are eating an oncosphere that is in muscle. So we're eating either undercooked pork or undercooked beef. And when we ingest the infected meat, then that oncosphere will essentially hatch. That little hook, as you saw here, this little thing is like the little baby scolex. It will release in the small intestine. And then, as a result, it will hook there, right? And then it will develop into an adult tapeworm. This takes time, right? This takes like months to get a tapeworm. And so when you get it, it takes a long time before you might even notice symptoms. And frankly, it doesn't feel like anything. If you have one, you might just notice the shedding of those proglottids in your feces, which usually tips people off, or, you know, sneezing one of them out your nose. That's quite unusual. I don't know how it crawled up the intestinal tract because, again, they don't crawl. They don't have like muscles that will allow them to crawl like that, so I don't know how that happened. Anyway, you ingest this Cysticercosis, which is kind of the structure that's in the muscle that has the oncosphere in it, and then depending on which species you have, you will develop an adult worm in your small intestine. What will then happen is sexual reproduction, or I guess, I mean it is because there's sperm and egg, but it's, you know, there's hermaphrodites, so they're fertilizing their own eggs. And the eggs or the gravid proglottids will pass in the feces. These can both be diagnostic. And those will get out into the environment, right? And so if the feces is outside, then cattle will eat it, and it will be the next stage of the life cycle. If it's solium, pigs will eat it, and they will get the next stage of the life cycle. And basically, when they ingest this oncosphere here, then they are going to, that little oncosphere is going to hatch, and it's going to penetrate the intestinal wall of the animal and get to the muscle, where it develops into that sort of protective structure, Cysticercosis. Now, like we saw with Diphyllobothrium latum, there is an alternate route of infection, and it's only for Taenia solium. But if we were to ingest the egg directly, so if you ingested the egg, so kind of like an auto-infection, right? Like you don't wash your hands when you go to the bathroom, you can develop Cysticercosis. And Cysticercosis is very similar to Sparganosis, although these are just different species, so they have different life cycle stages, so they have different complicated names. But Cysticercosis is when this thing that normally develops in the muscle of the pig or the cow develops in human tissue. And when that happens, if it happens in the muscle or in the brain or anything along those lines, obviously it's kind of dangerous because that will impact the tissue that it is in. I just wanted to make sure I told you the same stuff. It looks like I think I covered what I needed to for these big tape. These are big tapeworms. They can be like 26 feet long, right? Because think about how long a cow's gut is. Quite long. And if you don't kill that Scolex, then you don't kill the tapeworm. So, lovely. Let's take a look at our next case study. 28-year-old male from Algeria had complaints of right upper quadrant abdominal pain. As part of his workup at a medical facility, imaging studies revealed a complex solid and cystic lesion on the posterior right hepatic dome, so part of the liver, right? Hepatic cyst fluid was aspirated, so they went in with a big needle and they got some of the fluid out of this lesion, and they looked for OMP, and as you can see, this is what they found in a wet mount. So maybe pause and think about the organisms you've read about and sort of notice what you might think it could be. And then when you're ready, I'm going to tell you the answer. So pause for sure if you don't want to know it right now. But if you do want to know it, I'm going to tell you this is Echinococcus Granulosus. And what you're seeing here are structures called protoscolices. And they have these little suckers on them that you can kind of see in that figure A, like right here and right here. And these are unique organisms because they form, this is just normal in their life cycle, a cyst. It's called the hydatid cyst. And that was what this man had developed in his liver, was this cyst that was taking up a lot of space. And what happens here is you develop this big cyst, which then develops these little mini or daughter cysts. And inside of these little cysts are just little bits and pieces. So if we zoom here, little bits and pieces of developing Echinococcus organisms, which is kind of gross, but it's true. Now, a brood capsule, as you can see here, there's a couple here, are capsules in the cyst and sometimes in the daughter cyst that will contain those protoscolices, which is like the little early on scolices or scolex of the organism. Now, what will happen is when you do take an aspirate of the cyst, you'll suck up all this junk, right? That's floating around in this big fluid-filled structure, which I will show you. And that junk that you get is known as hydatid sand. So you're going to see older pieces, you'll see daughter cysts, you'll see scolices, you'll see hooklets. There's all sorts of nasty stuff that essentially gets removed when you do go in to look at the cyst. So please make sure you look at the hydatid sand slide. I think that we have one of those. Now, if you take a look here, this is the hydatid cyst, I believe, slide. And essentially what you're looking at here, so the green is the outer layer of the cyst. So if this is the outer layer, then this is the outer layer. And the yellow is kind of the germinal layer where the little daughter capsules and brood capsules sort of germinate, right? Blue, the dark blue, I think, is showing you the protoscolices that are developing. The black is a brood capsule that has some protoscolices in it, and the purple is pointing out the hooklets here. So the thing about Econococcus granulosus is this is a small worm. It only has a couple body segments, and for whatever reason, I don't think I have it shown here, but it's like 2 parts to its body. So it's little, but it's forming these large cysts in your body and in your liver oftentimes, and that can be really damaging to that tissue. It does have a rostellum, so you can sort of see here it is armored, and the eggs are identical to Tania. So this is another answer that you could put that would be correct for those kind of radial striations, so those little lines that are kind of radiating out. And then it has that oncosphere inside here. It's little, all right? It's little. What's not little are the cysts that are formed. Let me show you this picture. You're not even ready for it. These are cysts that have been taken out of probably a liver, but they could go other places too. And each of these would have different, you know, if you were to aspirate it, you would find that hydatid sand in there. So quite problematic. There are Echinococcus multilocularis strains that tend to form these multiple cysts, you know, rather than one big one. And granulosus tends to have a unilocular cyst, so it would be like just one big one without all these little ones. Now it's unique, this life cycle. It's kind of interesting because we have what we've got here is a sylvatic cycle, which is kind of like a wild cycle, and that's what you're seeing in the middle. So basically what would happen is a wild dog, think of a wolf, would eat some sort of intermediate host, sheep, goat, swine, that have this organism. And it sounds disgusting, but the way that these things live, they live kind of in your visceral organs, and when a wolf eats an animal, it sort of rips its guts out, right? And so when it does that, it's going to burst these cysts, and it's going to come into contact with all that junk that's inside of them. And so that's going to infect the dog, the canine, as you can see, or the wolf, and that will infect it. And then in the feces that it leaves, that will infect the ground that the grazers are grazing on, and the cycle repeats. I think on Isle Royal, the wolf moose situation actually is like this. And I think that there is, it isn't exactly this species, but it's something kind of similar. Now, I'm just going to read to you kind of the definition of this life cycle that the CDC gives. It says the adult Echinococcus granulosus, 3 to 6 millimeters long, resides in the small bowel of definitive hosts. Remember, definitive hosts are where sexual reproduction happens. Dogs or other canids. Gravid proglottids release eggs that are passed in the feces. After ingestion by a suitable intermediate host, natural conditions, sheep, goat, swine, whatever, the egg hatches in the small bowel and releases an oncosphere that penetrates the intestinal wall and migrates through the circulatory system to various organs, especially the liver and the lungs. So in this case with the human, what we're seeing here is we basically take the place of the sheep, and we ingest the egg that was left in the feces of the canine creature. And then because that oncosphere, as you can see, has those little sharp pointy things, it will go through your intestines and actually migrate to any of these places. All those number 4s are pointing to places where these could end up, essentially. Once it's in the organs, the oncosphere develops into a cyst that enlarges gradually, producing protoscolices and daughter cysts that fill the cyst interior. The definitive host becomes infected by ingesting the cyst-containing organs of the infected intermediate host. After ingestion, the protoscolices evaginate, that's a new one, attach to the intestinal mucosa and develop into the adult stages. So in the human, we would not have an adult worm. The diagnostic stage would have to come from an aspirate of a cyst in one of our organs. And the only way that we could pass this on is if some wild dog, or not wild dog, were to disembowel us, essentially, and eat the cyst, the stuff that's in the cyst. So the actual worm itself is going to be in the canine, and then we get the eggs from the canine and become the sheep, or the goat if you prefer, or the swine, either one, and we get that cyst in our liver, lungs, or wherever it might be. So definitely not great, and something that would probably require surgical removal. So we'll end here. One more video about the last group of tapeworms. I know you're excited.